Hello everyone, this video bacterial structure part 4 includes details of cytoplasm, nucleoid, plasmid, ribosomes, inclusion bodies, gas vacuoles and mesosomes of bacterial cell. So let's start. This is the structure of bacterial cell. In previous videos we have already discussed cell wall, plasma membrane and glycocalyx layer of bacterial cell. Now we are going to discuss remaining part of the bacterial cell. The cell membrane surrounds a complex solution referred to as cytoplasm or cytoplasmic matrix. It is a dense, granular or jelly-like substance. This chemical pool is a prominent site for many of the cell's biochemical and synthetic activities. Its major component is water. It is served as a solvent for a complex mixture of nutrients including sugar, amino acids and other organic molecules and salt. The component of this pool serves as building blocks for cell synthesis or as a source of energy. Cytoplasm holds another large bodies which are nucleoid, plasmid, ribosomes, inclusion bodies, gas vacuoles and mesosomes. First important component of bacterial cytoplasm is nucleoid. Definite nucleus does not present in bacterial cell. What does it mean? Membrane which can give defined shape to nucleus does not present around nucleus. So bacterial nucleic acid is devoid of nuclear membrane, thus called nucleoid. The nucleoid is a simple reason and it is not separated from other components of the cytoplasm by a membrane. Bacterial chromosome is very large compared to the cell dimensions, so it needs to be compacted in order to fit. The nucleoid is a region that contains the cell's chromosomes and numerous proteins. The chromosome of most bacteria are a single circle of DNA. Nucleoid is essential for controlling the activity of the cell and reproduction. It is where transcription and replication of DNA take place. Enzymes that serve as a biological catalyst also present a nucleoid which help the cell with replication. Proteins that have other functional and structural roles including assisting the formation of DNA, cell growth and regulating the genetic material also present a nucleoid. Apart from nucleoid, another genetic body present in the cell is plasmid. Plasmids are small, double-stranded DNA molecules that can exist independently of the chromosomes. Plasmids use the cell's DNA synthesizing machinery to replicate, but their replication is not linked to any particular stage of the cell cycle. That is why it is called independent genetic material. Both circular and linear plasmids can be present, but most known plasmids are circular. Plasmids have relatively few genes, generally less than 30. Many plasmids carry genes that provide a selective advantage to the bacterium in certain environments. Like conjugative plasmid helps a bacteria in transfer of DNA from one cell to another. Our plasmid, which is also known as a resistant plasmid, carry antibiotic resistant genes which give bacteria antibiotic resistance ability. There are so many other types of plasmid which give different benefits to the bacteria. Next entity of cytoplasm is ribosome. Large number of ribosomes present in bacterial cell. This is the site for protein synthesis. The type of ribosome present in bacterial cell is 70S. Bacterial ribosomes are called 70S ribosomes and are constructed of a 50S and 30S subunit. 50S subunit can further divided into subunit in 5S RNA and 23S RNA. 30S subunit can be divided into 16S RNA. The S in 70S stands for Swedberg unit. This is the unit of sedimentation coefficient, a measure of sedimentation velocity in a centrifuge. Sedimentation means settling in the bottom. The faster a particle sediments in the bottom, its Swedberg value or sedimentation coefficient become high. Or we can say heavier and more compact particles settle down faster and have larger Swedberg number. Beside earlier mentioned cell organelles, other important bacterial cell component is inclusion bodies. Inclusions are common in all cells. They are formed by the aggregation of substances that may be either organic or inorganic. Inclusions are used for storage. The quantity of inclusions used for storage varies with the nutritional status of the cell. Inclusions can take form of granules, crystals, or globules. Cells have a wide variety of storage inclusions. Some store end product of metabolic processes, stored material used by the microbes when they are in nutritionally deficient environment. The most common storage inclusions are glycogen inclusions, sulfur globules, and polyphosphate granules. Polyphosphate granules store the phosphate needed for synthesis of important cell constituents such as nucleic acid. Sulfur globules are used in energy conserving metabolic processes. Gas vacuoles are aggregates of enormous numbers of small, hollow, cylindrical structure called gas vesicles. Cells with gas vacuoles can regulate their buoyancy to float at the depth necessary for proper light intensity, oxygen concentration, and nutrient levels. Mesosomes are folded in vagination in the plasma membrane of bacteria. It is observed that these structures appeared when certain chemical or technique like electron microscopy exposed on bacterial cell. There is no known particular function of mesosome. So this was all about bacterial cell structure. Thank you for watching.